So we're going to begin by reviewing the physiological processes of the kidney and looking at the proximal tubule. So we said as we look at the physiological processes of reabsorption and secretion in the nephron, the processes used along the renal tubule of the nephron, we'll see many examples of primary active transport. This is a prime example. We'll see so many sodium-potassium pumps. The sodium-potassium pump is active transport as opposed to passive because it pumps things against their concentration gradient using the power of adenosine triphosphate, ATP. It pumps 3 sodium out of the cell, which is against its concentration gradient, 2 potassium into the cell against their gradient. So you can see the green and orange arrows showing the concentration gradients of sodium and potassium if they were diffusing. So we know it's active transport because it's going in opposite directions. Secondary active transport is also against a concentration gradient, but instead of directly using the energy of ATP like the sodium-potassium pump, it is driven by the ion's electrochemical gradient. So we're going to see many of these. The first 2 examples here on A are antiporters. That means that as sodium is diffusing back into the cell, it is using that energy of the sodium gradients to pump calcium in this first one and hydrogen in the second one against their concentration gradients. So it's against a gradient, it is secondary active transport. Antiporters because both calcium and sodium and hydrogen and sodium in these cases are being pumped in opposite directions. In the second one, notice as sodium goes back down its concentration gradient, it is pulling glucose and amino acids against their own concentration gradient. So it's kind of like they're riding the sodium gradient. In those 2 yellow ones, they are symporters because sodium and glucose and sodium and amino acids are going in the same direction. And secondary active transport, this is subject to transport maximums, which means we could have more glucose in the urine, for example, if we had an excess amount of glucose going through the renal tubule, if we had saturated all of our sodium glucose symporters. Someone that is diabetic that has very large amounts of glucose going through the renal tubule would expect to see large amounts of glucose in the urine, which would be abnormal and might denote uncontrolled diabetes mellitus. So it is subject to transport maximum. So obligatory water reabsorption is 90% of the water reabsorption we're going to see in the kidneys. That means that water is going to follow things like sodium, solutes that are reabsorbed, and there's about 10% facultative reabsorption, which means that it's just going to be absorbed according to the body's needs. And this is usually regulated by hormones ADH, aldosterone, and atrial natriuretic peptide. So we're first going to look at the proximal tubule, and the big thing you should associate the proximal tubule with is reabsorption. And as we've talked about previously, it reabsorbs 100% of organic solutes like glucose and nutrients, 65% of the water, which is obligatory as it follows sodium, absorbs potassium, 50% of chloride, and 80% of bicarbonate ions, HCO3, which is an important blood buffer. And it secretes hydrogen ions and urea waste, which is a byproduct of amino acid breakdown. There are also many microvilli located along the cells of the proximal tubule, and this greatly increases the surface area for reabsorptions, which is so important physiologically in the proximal tubule. A couple of the things that we're going to see in the proximal tubule, it says PCT here because sometimes you'll see it called the proximal convoluted tubule as opposed to the proximal tubule, but they're the same thing. We'll see a sodium glucose symporter, which takes sodium and glucose the same direction, glucose against its concentration gradient. Sodium hydrogen antiporter, which takes sodium and hydrogen opposite directions, hydrogen against its concentration gradient. And aquaporin 1 channels. Anytime we see an aquaporin channel, this is just something that makes water movement a little bit easier. It pulls and reabsorbs water out. So as we see in this figure, we see here on this inner membrane, we call this the sodium glucose symporter, as we are going to take glucose in through transcellular reabsorption. There are glucose transporters here on this outer membrane, this apical membrane we'll call it, and we can reabsorb glucose. And we've got sodium potassium pumps. By the way, about 6% of the body's ATP are spent just on sodium potassium pumps in the tubules of the kidney alone. Very significant here. We are going to see a little bit more of a complex situation with sodium hydrogen antiporters. Basically, all you need to know is as we take sodium in through transcellular reabsorption, we are going to secrete more hydrogens in the sodium hydrogen antiporter. So we get rid of more hydrogen as waste in urine. But it's produced when there is CO2 here inside of the cells lining the tubules, combines with water, makes carbonic acid. The bicarbonate ion is reabsorbed in the proximal tubule because we reabsorb 80% of those bicarbonate ions as they are an important blood buffer, and the hydrogen ion goes back in and is secreted. So the more CO2 that enters, the more hydrogens we secrete and the more bicarbonate we reabsorb. We also have some paracellular reabsorption, which means that ions travel between cells just by simple diffusion, and they can also go through transporters here, facilitated diffusion transcellular, and that's water, chloride, potassium, calcium, magnesium, and urea.